Okay, so conserved genes are a way of gaining confidence in the actual activity of an active gene. Meaning that if an active gene has a history, also within and between species, it gives you confidence in what it does, because you can correlate between the species and compare and see that if it has the same function uh, um, in all of them. So synteny um, relates to a comparison of the location of genes between the homologous of two species. Um, Such differences between genomes is prospect to evolution, um, and is called reshuffling, meaning that when you have differences between homologous genes and species, it means that it underwent reshuffling. Okay. Okay, you're referred to a movie, so enjoy the movie, the video. So for instance, um, mice have fewer chromosomes, only 20, right? But the organization and similarity is up to 99% homolog genes, and 96% are in synthetic regions. Meaning that although there's less chromosomes, the organization and the affiliation to homolog genes is very high. Okay, the mouse has the same number of gene families as humans, but less expression of each family's diversity of genes. So a lot of, well, I don't know if to say a lot is exact, but there are studies that have been done and still are being done with mice in order to produce different organs for humans. For instance, one of them is an ear. Okay, a gene that is not synthetic is more likely to be a pseudogene due to translocation from the original locus. Okay, so if we compare two species and compare their genome, that will allow us prediction of a gene's function if one of the species genes is identified. So that makes it easier, right? Because we can look for similar or identical sequences, axons, components of the gene itself, and compare between the two. Okay, so RNA lacks the ORF, and that makes identifying genes that code for RNA much more difficult. So when we want to identify those kind of genes, it's a lot harder because of the lack of the ORF. When we compare the genome of a mouse and humans, there is about 40% of differences between the two genomes. Where does this, where do these differences come from? Most likely from a history of deletions from the mouse's genome, meaning that theoretically the genome was bigger, larger, and parts were taken out, were deleted. It's estimated that our genome, the human genome, has about 30,000 genes. That's estimated, okay? All right. When we're talking about euchromatin, is the region of the genome containing active genes. The rest is called heterochromatin, okay, meaning that it does not have active genes in it. Alright, you're referred to see a movie. When we're talking about euchromatin, we have to remember that this might include information for structural of some kind of structural importance, right? We talked about that. So about 1% of the genome is accountable for protein coding exons. That's not much. In your head, you might say, wait a minute, that means that about 99% is junk DNA. Just remember that we talked about junk DNA, and that's a trap, because though some of the DNA can truly be junk, not all of it is, right? When we look at a gene itself, only about 5% of the gene itself is protein coding. So there's a lot of elements and a lot of components in the gene that are there in order to allow coding for protein. Okay. So till date, about 24,000 genes have been positively identified, and an additional 12K of potential genes. That sums up to roughly 36,000, right? And there's two different sets of analyses that have been done. The first estimates 30,000 genes in the genome, and the second one goes up to 40K. So there's not one definite number, but we're kind of moving on a scale between 30,000 to 40,000. But here's an interesting thing, fact. The overlap between the two was only 50%. Meaning that it's not, when you look at the 30,000 and the 40,000, it's not as if 30 out of the 40,000 were the same genes. There's only a 50% overlap, meaning that there might be much more than 40,000 genes in the human body, but we don't know for sure. Alright, so if we look at a RNA transcript, 11,000 genes were identified, accountable for the majority of the overlap between the two human gene sets. So most of that overlap comes from basically at the RNA level. Even if we have 50,000 genes, that would be relatively a small number in compared to our size, our body size. So it's estimated that it should be more than 50,000. Okay. Now we have to understand one thing. When we talk about even 1% of a difference between two genomes, that can be a huge difference when you look at the organism itself. When we're talking about the chimpanzees, how identical to humans are they? But they only have 1% difference between us and them. So it does make a difference. Okay, so in your head, if you're saying 1% is not much, it's enough to produce a substantial difference. Meanwhile, we are the ones going to the zoo and looking at the chimpanzees and not the other way around. Although the movie Planet of the Apes might think differently, right? Okay. Okay, so let's go back for a second to alternative splicing. And alternative splicing is a mechanism that may actually make additional genes unnecessary. So I'm going to explain that. If the theory was right and one gene meant one protein, then we would need a substantial amount of genes, right? But we know by now that there's alternative splicing, and that gives variation through the same gene. So maybe we don't need that much, so many genes. So this is, I know this sounds confusing, but we're kind of dealing and analyzing the different possibilities and trying to understand how many genes are there. On the one hand, we already identified between 30 to 40,000 genes with only 50% of overlapping. At the same time, we identified 11,000 genes at the level of RNA being the majority of the overlapping. And in addition to all that, we have alternative splicing that kind of gives us a lot more options to the same gene. Okay, let's continue. So, in the next slide, theoretically, one gene means one protein, right? But we know that's not true. Because in fact, one gene equals multiple proteins due to alternative splicing, up to 60% more. Alright, so alternative splicing is a major process that gives us a lot more outcome with a lot less genes. Alright, so when we look at the genome, it is full of what we call deserts. Deserts are long areas that have no genes at all. These can be regions of up to 50 base pairs, long. So when we take a chromosome that we will define as gene rich, meaning that it has a lot of genes in it, at least 10% of that chromosome is going to be full of deserts. 50% of the genome is sequences that repeat themselves. Okay, so you have, for instance, figure 3.24 on page 67, and you should take a look at that. And we can take those sequences and divide them into 5 classes. So the first class is transposons, right? The second class is process pseudogenes. The third one is simple sequence repeats. The fourth one is segmental duplications. And the fifth one is what we call tandem repeats. Okay? From all the 5, the one to emphasis and focus on would be the transposons. They actually may act as DNA elements, and sometimes actively even as RNA. And the question is, why are there so many transposons if most of them are inactive? You see, it doesn't add up. If they are inactive, why so many? May they have a role that we have yet to identify? Or perhaps their role is relatively to the values and the terms of the world of the field of evolution. So here are the options, theoretically. One, maybe they had a part in the past, okay, and they don't have it now, so they're just there, waiting to be deleted in some way. Maybe they'll have a part in the future. Remember, anything inactive right now might become active later, though we do not know the effect, right? Maybe option 1 and 2 are true. They're part of the past and they're still going to be part of future evolution. And maybe their true role has simply have been yet to be found. Meaning that they have a role right now as we speak, but we do not know what it is. There is evidence that transposons have evolved into genes and then moving their ability to move. So that gives us a hint that they might still have a significant role to play in the future. When we're talking about segmental duplication, that occurs all the time. But extreme case would be the duplication of a whole genome, creating what we call a tetraploid, meaning that those two sets of DNA information are duplicate, undergo duplication. Okay, we'll stop here and continue in the next video.